Good afternoon, everybody. Good morning, good evening, depending on where you are in the world. So welcome to the ECTRIMS hangout. So today we're in the Excel Centre in London, which 4 years ago was the site of the Paralympic Games, which is going on as we speak. And today it hosts many neurologists. So my name's David Baker. So speaking of blogging, while we're here, you can ask us any questions. We may be able to answer them, we may not, but please send them by Twitter. So the address is @BartMSblog. So that's @BartMSblog. So yeah, ask us whatever you like. We may answer it, we may not. Anyway, Sharmilee is going to tell us the what we're going to do today.
So I've structured this wealth of information which we've been this year. So the plenary lecture was all about the personalized medicine, what we should be doing in clinical practice today. And since there is now a massive amount of clinical trial data across the board from relapsing-remitting MS through to PPMS, I've decided to structure the hangout based on relapsing-remitting MS clinical trial findings, secondary progressive MS trial findings, and primary progressive trial findings. And then I'll hand over to my co-host to talk about the experimental treatments. And this is where some of the more idiosyncratic and interesting work is, in fact, can be found. And then we'll finish the hangout on biomarkers. Throughout, I'll be checking on the table about what information needs to be actually put across through to you. So if I'm looking away from my co-host, it's because I'm looking at also that information, but also I'll be looking to see what tweets you have put through to see if we can answer any of the questions you have.
I mean, it's been a really intensive meeting, and we will apologise. We can't actually cover everything. We'd have to have 10 heads and to be at all the sessions. Many sessions were running in parallel, and you know, it's been about treatment, and we probably won't be able to mention every single treatment. So again, if you want to hear about something, remember you can tweet us at BartMSblog. So over to you, Sharmilee.
So the plenary lecture was personalized medicine. So what do we normally understand by personalized medicine? Traditionally, it has always been based around your genetic profile. So some treatment is targeted to your genetic profile, or the side effect of treatment is targeted to genetic profile. The difficulty we have with MS is that it's a polygenic condition.
Polygenic means lots and lots of genes.
That's right. And when you look at the MS predictors, genetic predictors of prognosis, they're actually very weak and not very useful, and the studies have been contradictory. So Xavier Montalban, the leaving president of ECTRIMS, said that we should be discussing about the right drug, the right patient, at the right time, and at the right price. And that's very contentious, but I think more and more we'll be discussing that.
Really?
So, in fact, there's a couple of poor prognostic factors. One of them he mentions is the oligoclonal band status, which increases your odds ratio of developing CDMS by 2, and then the high EDSS score, as well as brainstem lesions, which you mentioned, and the new biomarkers, chitinase-like 1 protein, as well as the NFL light chain.
That's neurofilament light for you and me.
So he felt that the more we do personalized medicine, that we may not always be talking about NEDA, or no evidence of disease activity, but moving on to MEDA. Now that in fact stands for minimal evidence of disease activity. So we may not be able to achieve complete suppression of all MS disease activity, but we will get pretty close so that you have clinically no disease.
I say yes, we can.
So moving on quickly to the clinical trial findings, which have been exciting, I'm going to move on to relapsing-remitting MS, starting off with cladribine. So cladribine Professor G presented the clarity findings as well as the extension data. So, you know, from the first clarity data, the low dose 3.5 milligrams per kilogram dosing achieved a 58% reduction in annualized relapse rate. It also reduced time to 3-month confirmed EDSS progression by 33%. Now, what they did, interestingly, in the extension data is that they took those who were in the original placebo arm and put them on cladribine. And that was the most interesting data because it reduced their annualized relapse rate by 60%. So even if you were initially in the placebo arm for the first 2 years, you can still get very good relapse rate reduction later on. They also presented data suggesting that cladribine, oral cladribine, would be more of an induction treatment, and the treatment gap where you may not have to redose would be about 43 weeks. So if you wait for more than 43 weeks, there was re-emergence of durable of the MRI activity. So there was no durable clinical suppression. So that's cladribine.
So that's, I guess, the first question we have to say is this drug actually kind of fell away, and it was rejected by the European Medicines Agency in 2011. But I guess the data speaks that, you know, the environment has changed, and you know, there was a concern about the safety, and I think the data has really kind of shown that the safety issue that was a concern of why the EMA rejected it probably was a fluke. But at the moment they're going back to the EMA, and it may be a new drug. We have to kind of watch this space just to see what the regulators do. They may say yes, and that'll be one more drug, or they may say no. We don't know. But the data is looking good, and you know, it'd be nice to be able to have an induction therapy, which means you only actually take the drug for a short period of time, and then the disease, as you said, was about 55% showed no evidence of disease activity for a number of years. So rather than taking the drugs very frequently, then you know, it will be different. And one of the sessions was, should we go for early induction treatments, or should we try and escalate the treatment? So start off with a drug and see if it works or not, and if it doesn't work, escalate it to a drug with more potential efficacy but harder side effects. And it was interesting that the room was split whether you know, you should try the induction phase or the escalation phase. I guess this work boils down to where personalized medicine may be. But it was really interesting. One of the papers that was shown was saying from a registry in Sweden that if you look at treatment, it's better to be, if you treat early, you do better than if you treat late. So no matter what the treatment you are on, it is better to be on a treatment than on nothing. So
there's a question from the can I ask my neuro for cladribine, or can a neuro use cladribine off-label?
So about cladribine, it's the oral cladribine. At the moment, the answer would be no, you can't really have it, because until the regulators approve it, it won't be available. In terms of off-label cladribine, we did see a presentation of where cladribine has been used off-label, and it can obviously be as effective as the oral version, because the oral version just turns into the off-label version. But that then boils down to you know, whether the neurologist will be willing to do that. We'll talk about off-label a little bit in a minute when Sharmilee talks about rituximab. So
yeah, so rituximab the coverage today.
So rituximab is an anti-B cell agent.
Yeah, so it targets a molecule on the surface of the B cell called the CD20, and it's a mixed antibody. Part of it comes from the mouse, part of it comes from humans. And what they showed was amazing data, which that the evidence of any form of MRI or clinical activity was completely suppressed in terms of odds ratio to 0.01, which was phenomenal as far as I've ever seen. And these were patients going from fingolimod to rituximab, from natalizumab to rituximab. So amazing, amazing data. And definitely, I think B cells probably the hottest topic this year at ECTRIMS. Interestingly, rituximab, as you said, is off-label use in Sweden, and they had a distribution of a very interesting distribution of center usage. So the lowest they had was 6% up to 87% in one unit in terms of usage of rituximab. So definitely their knowledge around rituximab use is probably exceeds any other center.
And apparently, there was a complaint by a pharmaceutical company who makes, a same type of drug, and the Swedish government kind of discussed it, and they said, yes, you can do off-label prescription. So it will probably depend on country to country and neurologist to neurologist, but an interesting case. Ocrelizumab is also another drug that binds to the anti-CD20. This one is what we call humanized antibody, whereas rituximab has bits of mouse and bits of human, and it's called chimeric antibody. And so the humanized antibody should have less immunogenicity and should have less infusion reactions, although they do occur. But what was interesting, the metadata presented was 55%. After 1 year, and after 2 years, I think it was 80%. So, again, showing that, blocking B cells is a very, very potent way of suppressing multiple sclerosis, at least the relapsing multiple sclerosis. And, that was pretty good. And it's not necessarily the best thing. There are other treatments which have been reported. Yeah, so I mean, it's B cell. For many years we thought MS was T cell mediated disease. How does it work? I've asked many people. We're all kind of got our own ideas. One idea is it blocks the antigen presenting cells, which are going to stimulate the T cells, who will drive the disease, but other people will have other ideas as well. But talking about the NEDA rates, there was some work with hematopoietic stem cells, and the no evidence of disease activity was over 75% after 3 years. So blocking the immune system is clearly very important for the controlling multiple sclerosis. The issue with that type of therapy is we're still not clear about the risk-benefit ratio, because it can be dangerous, and so there was a big debate. Some people would argue that it should be reserved for people who are very active and who are failing the other diseases. We will see how that pans out, but it does tell us that it is possible to really get a good control of the relapsing disease, and we've got a number of drugs which are helping us do that.
So one of the questions going back to rituximab from the Twitter is rituximab an induction treatment, and is rituximab like ocrelizumab? So it is an induction treatment, and we should use it as an induction treatment. If you are looking to use it on a regular basis, you probably have to look at the risk profile associated with the drug and the heavy immunosuppression. So there is that risk of developing PML. So we do need to consider that carefully.
I think in the Swedish data that was shown, it was that most people on the rituximab weren't on it for many, many years. So if it is an induction therapy, means you have to take a few doses rather than take it all the time. It's going to be very interesting, because the pharmaceutical companies are planning to do ocrelizumab every 6 months. So, you know, and the idea is you do that forever, but you may not have to. So we'll have to wait and see what happens. But that's why the rituximab data is really, really exciting, because it does suggest that you don't have to treat forever. And that means you can be drug-free. I'm excited, yes.
Rituximab is like ocrelizumab, except its backbone is different. So as we explained, the original first-line rituximab is a chimeric, whereas ocrelizumab is more humanized to avoid any of the infusion or lower the risk of the infusion reactions.
And ofatumumab is fully human, so it hasn't got any mousy bits in at all, and so the potential of infusion reactions should be even lower. So.
Right, so we're still on relapsing-remitting MS, David.
Yes.
And I know that our bloggers always ask about alemtuzumab, so I couldn't completely ignore it. But there was the 6-year data on CARE-MS1 and CARE-MS2, which is positive again in brain atrophy. But what I was more interested in was the adverse event profile or the side effects of the drug, really. And what they showed is at 6 years, you're looking at 42% of patients experienced a thyroid adverse event. Interestingly, this happened in the first third year after the infusion, and then steadily went down. And in terms of serious adverse events, it was also at year 3 rather than very early on. And unlike most thyroid problems which you see de novo, the type of thyroid adverse event was more skewed against hyper, that is an overactive thyroid, rather than hypo, which is an underactive thyroid. So something different there with what the drug is creating. Now, the other one which everyone's always interested in is the ITP, the effect on platelets. So platelets are also called as an autoimmune disorder.
They're the ones that make blood clot, I think.
Yeah. And that falls to 2.6%. So it's a rarer, much rarer side effect. And then the kidney abnormality called Goodpasture syndrome or antiglomerular basement membrane disease, it's the new figures are 0.2%. So if there are clinicians watching this, these are the up-to-date figures on alemtuzumab risk profile. So so moving away from relapsing-remitting MS, we come to secondary progressive MS. Now, we know that there have been negative trials on this, one of which is the ASCEND trial, which is natalizumab. This was a phase 3 study which on compound scores for looking at disability progression at using EDSS score, PEG test, as well as the time 25-foot walk was initially reported as negative, David. And what is happening with this is this year they presented the subgroup analysis on upper limb function. So exciting data on this, because there is actually a significant protective effect of natalizumab on upper limb function in regard to 9-hole PEG test performance. And in fact, those who did better were in the higher EDSS score category, so were more disabled with an EDSS score of 6. And also, they didn't have many relapses or hardly any relapses in the 12 to 24 months preceding the duration of the trial, and this finding was independent of GAD enhancement. So very interesting data, because we always consider natalizumab to be anti-inflammatory. So if we believe in the focal inflammatory model hypothesis, maybe this doesn't quite back it up. So that's secondary progressive MS, but we've also got primary progressive MS. Now, everyone's heard of ocrelizumab, or the oratorio trial, which claimed most of blog space all of last year with its positive data. It showed a 24% reduction in 12-week progression in disability and a 25% reduction in 24-week clinical disability progression. Now, what they presented this time around is term of no evidence of progression, or NEP. Very convenient, I think. But what they demonstrated is that ocrelizumab demonstrated evidence of NEP in 43% versus 29% in the placebo arm. That is a 47% relative reduction of no evidence of progression with ocrelizumab in primary progressive MS. So what the question I have is, when are we going to get this drug in clinical use? Because this is clearly an unmet need.
Yeah. Okay. Well, I guess, again, we're just waiting for what the regulators have to say. I think there's some other really nice data as well presented where, we've been talking about hands quite a lot, actually, at this meeting. And it was shown, actually, that if you look at the rate of progression of the lower limbs, although the drug actually was pretty good and had an effect on slowing disability of the lower limb function, actually the effect on the upper limbs was actually even more startling. And I think, obviously, there's been a little campaign run about getting a focus on hands as being an important thing that we really need to think about when we're doing our clinical trials, because hands are very important for people, as you know, and for many times the focus has always been on the EDSS and the mobility. And we had a burning debate, which was quite challenging, about whether we should be treating people in wheelchairs, and it was quite an entertaining session. If you want to watch it, you can watch it on the ECTRIMS website. I tell you, it was. Whoa.
So David, just to break you away, I'd like to say there's another very interesting question, which is, how do drugs in relapsing-remitting work in secondary progressive MS?
Woohoo! Well, I think that's always a difficult one, and it's a question I also will be putting. So I think one thing that is very clear is that people with secondary progressive MS, it's part of a spectrum. You don't go from relapsing remitting one day and secondary progressive the next day.
I think completely agree with you. The inflammation, as far as your EAE works, it's always there, right? It doesn't disappear on one day.
No, and that's what the pathology says. If you look, you can see, the similar things that you can see early in MS, late in MS. But we work on like animal models, and that EAE is experimental autoimmune encephalomyelitis. But there, what we can do is we know that the inflammation causes relapsing-remitting disease, and then it will transfer into a progressive disability. And what we know is that we can stop the relapses by blocking the immune cells, the T cells, from driving the relapses. But once the slow progression starts, there's something different, we believe. So I think we do still need different treatments to tease out the progression, but I'm pretty convinced that underlying some parts of the progressive disease, that there is an inflammation going on, and we really need to do that. I mean, one of the problems has been is some of the drugs are very expensive, and therefore when you're doing cost benefit, some places such as in the UK, they say it's not cost effective. It doesn't say it's not effective. And I'm sure that elements of progressive disease will clearly respond to these types of drugs. And I think the data we have from the ocrelizumab, which has been presented, and natalizumab, tells us this. So there are drugs that work, and I think we know from the animals is that the inflammatory disease kind of conditions the progression. We don't know if they're two completely independent effects, but again, with the animals, you can see if you actually deal with the relapsing attacks early, then the progression either doesn't occur or it occurs very, very much later. So again, it's speaking that we need to get inflammation under control if we're thinking about other things. Now, how does progression work? I mean, there were some really interesting sessions looking at mitochondria. Mitochondria are the cell, the energy factory of the cell. and they're very important in helping a nerve signal, because actually the brain uses most of the body's energy. So, you know, not very big in my case, but it uses about 20% of the energy. And so in MS, when you have the demyelination, it makes the nerves work much harder, and this requires energy. And some of the studies that were showing would actually that. So there was some experimental stuff where they have a mutation in the mitochondria, so they couldn't make too much energy, and actually not that much was happening. Slowly these animals would actually get a progressive disease, but what they showed is when they really stressed the system and made the, you know, lots of activity for the nerves, then actually they conked out a lot quicker. So, you know, telling us energy metabolism is potentially one of the things that we may be able to target, and hopefully that will then help save the nerves, and maybe, you know, that will be part of the equation, because we've got ideas about how to target those things, and some of those we kind of discussed. They're a bit, you know, science fiction at the moment, because, you know, some of those drugs are a long way off, but we'll come back to some of those as well a bit later on as well.
So we have another question, an equally good question. So someone who knows what they're talking about. Do the new drugs get to the CNS? Well, yes and no. Rituximab is a classic example. So the NIH had to do a clinical trial with the intrathecal rituximab. That's
kind of doing a lumbar puncture.
Yes. So you do a lumbar puncture, and then you put the drug directly into this fluid space which bathes the brain and the spinal cord. Interestingly, what they found was there was quickly suppression of B cells. So rituximab is an anti-B cell treatment. There was suppression in the bloodstream very quickly on, but then the cells came back in the brain. So
I think the problem is like the flow kind of is down, and probably I've heard of a study where they may think about actually trying to deliver it up into the brain to wash it out, but they say antibodies don't necessarily get into the brain. But we can ask the question, do those antibodies need to get in the brain? I think for many things, actually, no is the answer. Certainly from the animal work, we know that the relapses are caused by the cells going from the blood into the brain. So if we stop them in the blood, we don't have to worry about them. Yeah, and fingal in the same way.
So we've got another question, which is, does exercise damage mitochondria? I don't know, what do you think?
I don't know the answer. I don't know. I mean, obviously, nerve function requires energy. It will make the mitochondria work harder. But the question is how hard do we need to drive them, you know, the energy deficits? And I don't know the answer to that, but it's a really, really good question.
I mean, there's definitely
yes.
And reason cardiovascular. So there is clearly some kind of probably a boosting function, would you not say, on the mitochondria through exercise? But how would you test that? I mean, that's
well, it'd be an interesting one to do. And, you know, at the moment we're seeing some, you know, of the Paralympians getting their exercise and they're getting the gold medal. So, I mean, exercise, I think, on the whole, is generally good. Whether too much of a good thing is bad for you, I don't know. But
right. So moving on from the actual clinical drug class, we're now going to move on in the latter half of the blog, hangout, to experimental treatment. So there's some wacky data presented in this section.
Well, I mean, first of all, I'll start going back onto that energy thing. And one of the ways of affecting the energy is by the balance of the ions inside the nerves. And a few years ago, you know, we and other people came up with the idea of blocking the accumulation of a certain type of ion into the nerve, and it would help the nerve not have to expend so much energy. And in doing so, we would predict that maybe it would slow down nerve damage. So that's the idea. And today, or, you know, a few days ago, we actually heard, well, actually the idea can translate. And in fact, if you block sodium channels in the nerves, then you can potentially slow down nerve loss. So that's a good thing. So it's telling us that we can actually start to target elements that we believe are occurring in progression, and and that's independent of the effects on T cells and B cells. This has got a completely different mechanism of action, and so that was really exciting news. And so that was also then tempered by some unexciting news, or sad news to some extent. So phenytoin blocks sodium channels, and they tested it in optic neuritis. That's inflammation of the optic nerve. And
so they had an initial loading dose, and then they followed it by a lower dose.
Yeah.
For 3 months.
Yeah.
And then took the observation period up to 6 months. And it's an interesting trial design. When we talk about clinical trials and progressive trials, they've always been quite tricky. And one of the things to decide is how long in optic neuritis is that inflammation episode, and is that what's leading to the axonal injury? Now, a lot of the OCT data has OCT, which is an optical coherence tomography, which looks at the nerve fiber layer at the back of your eye. They demonstrate that the nerve fiber layer loss occurs within the first 3 months. So that's how Raj Kapoor, who's the PI of the study, targeted.
That's not a private investigator.
Yeah.
Principal investigator.
Principal investigator. Targeted this clinical trial. So he gave the phenytoin versus placebo for the 3 months, and then looked at retinal nerve fiber layer thickness, and that was positive finding last year. It showed that there was neuroprotection, and later on there was additional data work done by our group, by myself personally, which showed that the neurofilament levels dropped during the first 3 months as well. So
that's amazing, because that's really saying there is protection of nerves, because as the nerves get damaged, they kind of liberate the content, and Sharmilee was able to pick that out in the blood.
In the blood. And I think the consistency of these findings, first the lamotrigine study in secondary progressive MS, now phenytoin optic neuritis, the consistency gives us greater confidence in this type of methodology of blocking sodium channels.
Yeah, I think that's the exciting news. It is confirmation using a different technique, and a second study that kind of tells us we're onto something. And we are actually looking at that at the moment in a trial with another sodium channel blocker. So let's really keep our fingers crossed that we're really onto something. Now, at the same time, it was a bit sad because there was another study called a drug called amiloride. Now, this affects a different type of channel, but does something similar. And the data in the animal models was very good, but the trial was done, and the trial again in optic nerve, and this time it failed. Now, why? Now, one, you could say it's not the drug, but I don't think that's the answer. I think the answer was in the trial, and the difference was that in the amiloride trial, it took up to 28 days for people to get onto drug. Right?
So I think the window is 2 weeks.
Yeah.
Which is the phenytoin study, and we discussed more in greater detail than the other clinical trials we're going to discuss, but really under 2 weeks is what you should be aiming for neuroprotection in optic neuritis. But what about the visual evoked potential data? So VP is the latency of conduction of vision light getting to the back. That was bizarre. Do you want to explain to them what that was?
Well, that improved. So that was the suggestion.
It got worse.
Slower.
Yes. So there was a prolongation of the conduction of visual input into the back of the brain in the amiloride group. I think the delay was 27 seconds in the amiloride group versus 17 seconds in the fellow eye in the placebo group. So this unexplained prolongation is bizarre, because it's telling us that the amiloride's making. And there were a couple of hypotheses which the presenters put forward, whether in fact it's actually, this is a acid sensing channel blocker called ASIC1, and whether this is leading to a reorganization of ion channels in the optic nerve, and whether this is also present in oligodendrocytes, this channel, whether that's actually blocking their recovery. So there is some worrying findings in the right.
Well, I guess the truth will be out, or we'll find out, because I think, you know, amiloride is one of the drugs in MS SMART, which is a trial with 3 different drugs, amiloride being one of them in secondary progressive MS. Now, there is another drug called Prozac of fluoxetine, and that's also one of the arms. Now, we don't know what the result is, but tomorrow there's some late breaking sessions, and normally they don't put negative trials in those things. So actually, you know, we could be onto a winner there, but you know, we'll have to wait and see till tomorrow. But that's part of the exciting stuff that we'll get tomorrow, about whether fluoxetine or Prozac actually is beneficial in progressive MS. So we'll find out about that tomorrow.
So we've got 2 other questions about sodium blocking. One is, can I use sodium blocking drugs for MS? Yes.
There are common drugs used in epilepsy. There's one of them.
And a lot of your neuropathic agents, so the ones where you have tingling and drugs get given, they also, some of them block sodium channels. So you can be taking it for your MS without realizing, but not for neuroprotection purposes, because all of this evidence which we're presenting here is what we call phase 2 trial evidence, which isn't a large study with thousands of people. So phase 2 study only has about 60 patients or something like that, up to 250 patients. Whereas a phase 3 is much larger study, and that's what's needed.
And we got to remember that these drugs don't come without no side effects, and you know, so that is one potential problem. When you block the sodium, you're blocking the nerve excitation, so it can have, you know, undesirable things as well. So it's got to be a balance.
And then there's the second question, which is, did they use steroids with phenytoin? Yes, they did use steroids. But Raj Kapoor also presented data evidence saying that steroids don't actually have a major improvement on the actual axon.
I think what happens is it might kind of make the recovery a little bit quicker, but it doesn't really impact on the kind of the nerve damage per se that's occurring. So I think, as Sharmilee said, people were put on steroids because that's the standard treatment. So everybody got standard treatment plus the option of having phenytoin or not. And I think the amazing thing that the team did is that I believe they got most of the people on drug in less than a week. You know, so this again may be the key why phenytoin was useful. So you know, speed is of the essence, and we know that is true based on our animal work. So you know, it's the key is getting, you know, treatment for the optic neuritis as quick as possible. Now, in reality, in MS, I think it's different because the lesions are coming and going, and so you know, you've got more time to actually start the drug. You know, it doesn't mean that you have to get people on drug within 5 days of being diagnosed with MS. It's because what you're doing is you're targeting the effects of the lesions as they come and go. So you can start your treatment whenever you want. So it's not a question of, you know, getting within a few days for it to have to work.
So we've got a question here from MS tonight. They'd like to know whether a good amount of MS patients he encounters talk about mitochondrial Q. Is this just silly?
Well, it's not silly. I mean, it's all about evidence, and you know, we're not going to start saying mitoQ is great, mitoQ is rubbish until you actually have the studies, and the studies haven't been done properly. So it's a question of, you know, the proper studies have to be done before we can suggest that they're good. It's not to say that they're not good. It's just that hand on heart. We don't have the evidence to suggest it does work or it doesn't. But you can see there is a logic of why it may be. But you know, it may not be the whole answer. So again, we can't say, yes, this is a thing that you should be doing. Likewise, we don't say, no, you shouldn't be doing it. We'll just have to wait and see until the studies are done. But, it's not a silly idea at all. Good question.
Right. Before we move away from sodium channel blockers, we've got another question. Should you take phenytoin for relapses? Well, I think, the data is definitely supporting it, but if you want to be absolutely certain, we need a much larger study. So I think we should be lobbying for these clinical trials which are showing very good data in the first instance to be reproduced as large studies.
I mean, that's actually the type of experiment that the optic neuritis trial suggests. It says it should be during a relapse is where the drug could be useful. So we just have to try and do those experiments, and that's.
Now moving on swiftly to Lingo-1 or opicinumab.
Oh, I don't know. I don't know.
To be studying relapsing-remitting MS.
Yeah. My concern is I might not have to learn the name of that one because, again, this is a drug that was tried in optic neuritis, and it was also tried in MS, and unfortunately, the data is negative. And what does it say? Does it say Lingo-1 is no use for remyelination? No, it doesn't say that. The Lingo-1 was targeted with an antibody, and much of that doesn't actually ever reach the central nervous system. But it was shown that some did get in. Okay.
They did spinal sampling, and I remember somebody asking, do we do lumbar punctures in F1? So I'll just quickly answer that, yes. And then moving on swiftly.
So yeah, volunteer for a lumbar puncture whenever the clinicians want it, please.
Now the going back to the Lingo, so they did do spinal fluid sampling, and they showed that saturation of the receptors in the CSF. So that's that. So it's getting in there. It's working, David.
So they kind of were trying to tease out their data to say, well, can we get any positives out of that? And I guess what they were saying, and I don't know whether this is a positive thing, is if there were to be any effect, it was largely in people who were younger in terms of having a shorter disease duration, who hadn't accumulated as much damage. And so that's kind of...
And in the relapsing.
And relapsing-remitting. So it's probably not the news we want, because I guess people with secondary progressive are thinking, that's where you want the remyelination, but it's going to be much easier to actually remyelinate something early rather than later.
Yes,
and
if anything drops out from the clinical trial, but they didn't control for the multiple comparisons. So I'm not really convinced that, in fact, that any of the factors you listed, in fact, are important factors, because if you correct for that in most statistical models, then it may not be significant, and we need to see that data.
So, I mean, I suppose it was, essentially it was a negative study, and that's the take-home message, unfortunately. But not to say, we're not going to go back and try. We learn from our mistakes, and there are a number of other studies that are ongoing. I heard of a target called retinoic acid X receptor, or RXR, and there's a Cambridge group, and we heard that the study will hopefully be starting in the early new year. So that one is ongoing, and we heard a company say they've got an RXR drug, and it was looking pretty good in the preclinical models, but whether they materialize into benefit, we don't know. There are a number of other clinical studies that are actually ongoing. We just have to wait to see the results, to see if there's any benefit. And I mean, how many studies did I show showing new targets for remyelination? More than I could shake a stick at. It was loads. So this is another thing of, watch this space. It's starting to happen. So I can be optimistic about that. It won't happen quick enough, but that's the nature of science.
So the questions on Lingo-1 is Lingo-1 drug for improvement of which areas? So the clinical trial was weighted towards 20% of patients actually had secondary progressive MS, and the other half had relapsing-remitting MS, but the secondary progressive MS were early secondary progressive MS rather than later. So the major problem with this trial is there's no actual outcome measure. It was all clinical composite scores.
Yes.
And so you don't know where the drug was actually working. So clearly there needs to be a requirement for remyelinating and regenerative biomarkers, which is something to think about. So this draws a major point, which is can you do a clinical trial without having relevant biomarkers available?
Well, it makes. I mean, I think this has been, one of the lessons we learned from the Lingo trial. It's really what is the kind of the optimal way to measure a positive outcome, and that hampered us for a long time with relapsing-remitting drugs, and then we had MRI came along, and we had gadolinium, and it gave us a really good kind of way in to get a suggestion, and we're actually at early stages of those things. So there was a number of imaging studies suggesting new potential markers for monitoring progression, a repair, but we have to again put them to the test to see if they work or not.
The second question was anti-lingo drug in association with the DMT. Yes, that was correct, in fact, because the trial was on top of interferon beta 1A. So you have to stop the inflammation first. So if you're going to do neuroprotection, remyelination, regenerative treatment, you've got to get rid of the inflammatory cells, right?
Yeah. I mean, I kind of think about what you're trying to do is put wood onto a fire. Unless you put the fire out, you're just going to fan the flame. So I think the most logical and sensible thing is to control what we know we can control, and then we can either if we control it early enough, maybe the problems don't start in the first place. If not, we get control of what we can control of, and then we try and repair and replace. Would be the most logical thing. We'll have to see that happen in practice. So again, I think if you try and do a trial where you haven't got the inflammation under control, you're just making it so much more difficult to actually find a positive response. So good question.
So moving on to the last final leg of our blogging, it's about biomarkers really. So at the late breaking news, thank you David, is more data on neurofilaments. So I haven't a clue what's being presented there, but it's the light chain by Jens Kuhl from Basel. And but what was interesting today was about JCV index and JCV biomarkers. Can you predict PML risk? Now, we know that Biogen has introduced risk stratification, but despite the introduction of risk stratification with the test looking at the serum index, index level, there have been increasingly more case accrual. So risk stratification doesn't seem to be working, and we do need better biomarkers. So there was a good presentation which looked at various other predictive biomarkers. So there's something called lipid-soluble IgM oligoclonal bands, which is usually reflective of highly inflammatory MS. And what they found is that the risk of PML development reduces in patients who are positive for this. Obviously, this is a single center study done by VILA, and we need to see if others can reproduce this. And then you've heard of body weight. Body weight is also a risk factor. Age, and more interestingly is the L-selectin, or what's called the cluster differentiation factor 62, which sits on the surface of CD4 lymphocytes. And in that, they showed that if you combine it with the JCV index of greater than 0.9, 9, a low level of L-selectin was increased your risk of PML by 55-fold. So definitely a biomarker to watch.
It's an interesting one. So L-selectin is a marker that helps white blood cells get into the lymph glands. So again, it fits with the whole story about natalizumab affecting migration of white blood cells. So that's another.
So interesting you should mention that. That was another thing which that index levels have been demonstrated by more than one group now to rise with natalizumab use, suggesting that there is some form of immune system activation which is occurring in the background with natalizumab use. So your index could be creeping up on natalizumab. So I mean, so as you can see, there's been tons of information at this year's ECTRIMS, which is just the most exciting thing. I don't know, Dave.
Well, there's going to be one more exciting thing. Oh yes.
Okay, yes.
And we don't know what the answer is, unfortunately. But tomorrow, we know that data is going to be presented of siponimod, which is another sphingosine 1-phosphate modulator, like fingolimod, the brother of fingolimod or the sister. And this has been tried in secondary progressive MS. And we know the headline results, and the headline results are it is positive. So that's absolutely fantastic news, because that is the first drug in phase 3 that has had a clear effect on secondary progressive MS. So really interesting.
So many spin-offs. Okay, so the fingolimod non-selective, but now we've got other companies, Celgene, which has produced ponesimod and osinemod, which are selective SN1P blockers. So watch this space. The treatments available for MS are going at an exponential pace, really.
So the question is why did siponimod work when fingolimod didn't work? So
we don't know that data yet.
No. So we can say the primary endpoint is positive because it has been announced by Novartis. So we don't know the whole story, but we do know the headline result is positive. Now, I am going to be cynical. Now, the question is we know that fingolimod works by blocking relapsing disease. So the first question I will be asking is, were people in the trial, people kind of possibly a lower EDSS score who had active MRI lesions? Because if they did, then I would predict that that element would be inhibited. And if that element would be inhibited, then there's a chance that it would then impact on the accumulation of disability. So it could be that it's, the way that the trial has been done. I don't know. We'll have to wait and see. Yeah. Now, there was some more data presented today looking at whether cells could cause remyelination, and there was some data presented that siponimod was stimulating oligodendrocytes to cause remyelination. So it may be doing something different to fingolimod.
Neurofilament levels as well. So, the freedoms analysis showed that there is some axonal protection of fingolimod. So these are all interesting developments.
I think we also have to be careful when we say trials are negative. It's all relative. So Ascend, we said it was negative, but if you looked at the hand data, the hand function was clearly protected. So it's not always negative. The problem is when you do a clinical trial, you have to say before you do it what it's going to work on. And if you say it's going to work on the EDSS and it doesn't work, the trial fails. If you say it's going to work on hands, then the trial is a success. So that the reason why they do that is to stop people fudging it and picking and cherry picking what the results show. So trials can be kind of positive when the world claims they're negative. It's just how you look at the information, and so we'll have to watch the space. But I'm really, really excited.
So I think, you know, to bring everything to a close, I think the most interesting thing has been, I mean, what has been your highlight? I know for myself personally, it's the rituximab data. B cells.
B cells, yeah. And how does it work? I mean, that's going to obviously get scientists working for many, many years. And the question is, you know, can we do off-label prescribing? Who knows? We'll have to wait and see. But that's more or less all we've got time for today. So I'd like to thank you very much for joining us in what was very sunny London this week. And well, thank you. And thank you for your great questions. They were actually really good questions. Yeah.